Thank you for coming back for day two. Nice to see some familiar faces in the audience, and welcome to everyone that's here online today. So what a great day yesterday. So round of applause for all the speakers, everyone that participated in the family forum, the panels. It was a really, really great day yesterday. I really took away from myself personally a whole page of notes. A lot of the content I found was just great life advice also. Not just about Duchenne, but also just about life, and really touched me, and I really appreciated being part of that. So today is Sunday. So again, thank you for joining us for day two of Defeat Duchenne Canada Family Forum, hosted in partnership with Children's Hospital of Eastern Ontario. For those who couldn't attend yesterday, my name is Brian Atkinson, and I'm the chair of the National Board of Directors for Defeat Duchenne Canada. Just a few reminders for those who are joining us online today. If you feel comfortable, we encourage you to turn on your camera so that presenters can see you and interact with you throughout the sessions. Your audio has been turned off, and we request that you keep it disabled as you participate, just to reduce any background noise. Your keyboard will become your voice, and we encourage you to add your questions to the chat feature in Zoom. And this worked really well yesterday, capturing those questions that the online participants had. We want to ensure you can hear the sessions clearly. So if you'd like to test your audio, please click on the up arrow next to the audio button on the bottom left-hand side of your screen, and this will allow you to adjust the volume level. And there's instructions there on the slide for how to do that. For everyone online and those in the room, we encourage you to continue to use our mobile app, Hugh, on your computer, cell phone, or tablet. I was on the app yesterday. It was great to see people participating online from across the country, and it's a really, really cool app. So there's lots of pictures being posted of people's children and families, and then pictures of the event. So again, continue to use the Hugh app today. You can add questions in the Q&A sessions. You can participate in sharing the community discussion boards. You can create one-on-one and group conversations through direct messaging tool. And don't forget about our great contest that we have. So every time you use a feature on the Hugh app, you will earn points. The participant with the most points by the end of the weekend will win a $25 Amazon gift card. And the second contest is all about selfies. So those that are here in the room and everyone online can join, upload a selfie of you participating at the conference, and the photo with the most likes will win a $25 iTunes gift card. And bonus points if you're sporting your Family Forum t-shirt, as I am today. So if you have any technical difficulties with the Hugh app, email our coordinator of administration and donor relations, who's sitting at our tech table, Kate. Kate's over here. Morning, Kate, just drinking her Timmy's. She can help you out if you have any questions. Or you can also email her at kate.foster@defeatduchenne.ca, or you can also direct message her through the Hugh app. Also a reminder that some of the sessions for today will be recorded, and you'll receive an email to access the playlist in Hugh and our YouTube channel later this week. We also did record some of the sessions yesterday. Obviously, some of the sessions were sensitive, so we didn't record all the sessions because there was personal conversations, but we did record some of the sessions that you can enjoy if you missed them. We encourage everyone to follow us on social media at Defeat Duchenne today and throughout the year as we share the latest information and resources for our Canadian Duchenne community. And I was on Instagram this morning while I was having a coffee. It was nice to see some of the posts from yesterday. It was like, I said, Lisa, I saw you up on the stage yesterday on Instagram this morning. So it was nice to see our new CEO on the social channels. And finally, your feedback on this hybrid event is critical in helping us plan future family forums. So at the end of today, we'll ask that you take 5 minutes to complete the evaluation in Hugh under resources, surveys, and evaluation. Next, I really would like to just reiterate our gratitude to our partners at CHEO for helping us host this conference this year and hosting us here in Ottawa. Every year, we co-host a family forum with a leading neuromuscular care clinic. And again, a big thank you to the CHEO Hospital, and specifically Dr. Leanne Ward and her team for helping to create and deliver this weekend's amazing program. So let's take a moment to give them some recognition, please. So thank you, CHEO. And next, our list of great sponsors. So our education and support programs would not be possible without the funding support of our educational sponsors, as are listed here in our slide. A special thank you to our platinum sponsor, PTC Therapeutics, our gold sponsors, Dyne Therapeutics and Edgewise Therapeutics, our silver sponsors, Itel Pharmaco, Pfizer, RegenXBio, PepGen, Avidity Biosciences, and Roche. Our bronze sponsors, Solid Biosciences, Wave Life Sciences, Sarepta Therapeutics, and Biomarin. On our print sponsor, M&T Printing Group, you can get to know our sponsors by viewing their sponsor profile on the Whova app, connecting via direct messaging, and asking questions during today's research and drug development updates. So let's get on to today's agenda. So we'll be starting our day off strong with a comprehensive overview of the treatments for Duchenne and Becker, recent discoveries, research and progress, accessing clinical trials and approved treatments in Canada, the challenges being faced and the rare disease strategy being implemented to address these specific challenges. Then we'll take our first deep dive into the science and research developments from our industry partners at Pfizer, Roche, ReveraGen, and Dyne Therapeutics. Then we'll take a quick break for some coffee and snacks. And then we'll return to our second deep dive into the science and research developments from industry partners PepGen, Avidity Biosciences, RegenXBio, and Edgewise Therapeutics. So today's a great day to get some updates on treatments and access to treatments in Canada. So the next part to me is very special. I get to introduce John Davidson, who is the founder of Defeat Duchenne Canada, formerly Jesse's Journey. So you'll hear a lot about the fantastic research and developments in Duchenne and Becker science today. Again, as I said, our clinical trials and medical development. But first, let's go back to a time when there was very little known about this disease, a time when there were few clinical trials and a lack of funding for international research. To help paint the picture where we've come, I'd like to introduce the founder of Defeat Duchenne Canada, formerly Jesse's Journey, the fantastic John Davidson, joining us virtually.
Morning, Brian. I hope you're able to hear me.
We can. Good morning.
Good morning, Brian, and good morning to everyone in the room. I want to welcome all those families who are joining us today. And I've been asked to speak just a little bit about Jesse's Journey, which has now become Defeat Duchenne Canada, and to talk a little bit about how we came about and how we have grown to become Defeat Duchenne Canada. And for all those families that are with us today, you know, there's a little boy at the heart of everyone's story. In our family, our son Jesse was born in 1980. He was the second of our three sons, and he was diagnosed with Duchenne when he was 6 years old. Like all other Duchenne families, our world had just flipped upside down. And when that shockwave passes over you, I can tell you, as you probably already know, that first you cry. And then you ask yourself a lot of questions. Questions like, why us? And what did we do wrong? Eventually, you come to realize that despite what has happened, what's needed is action. Like a lot of moms and dads who are with us today, we knew nothing about Duchenne muscular dystrophy. And all of a sudden, we had been plunged into the fight of our lives. And we were going to have to fight on two fronts, I discovered. I came to realize that not only was there very little funding in place, there was very little research to fund. The field was very small, and there were very few players. As a dad, I asked myself this question: how does my buying another wheelchair help to solve this problem? The answer was, it doesn't. We needed two things. We needed more players to enter the field, and we needed the dollars to fund the research that we saw as the only route to finding a cure. It took some time to examine the landscape and to figure out what we were going to do. It didn't take very long for us to determine that we were going to have to do something sizable, something that would capture people's attention and fuel their imagination about what can be done when you show people that quitting is not an option when you want to make a difference in the world. And so on a cold morning in May of 1995, when Jesse was 15 years old and I was 49, we set off together as I began pushing Jesse almost 4,000 kilometers across Ontario with the sole purpose of raising money to fund research. For 124 days in a row on the road, starting with those lonely days in Northern Ontario, Jesse and I told our story every day about the need for research. Most people said they had never heard of Duchenne. Those same people were shocked that Duchenne affected boys almost exclusively, and that the disease is fatal and there is no cure. Day after day, we pushed on, and with each passing day, Jesse's journey was gaining momentum, and so was our ability to begin funding research. The message about the value of research resonated with people across Ontario and beyond. And day after day, we continued to tell the story, and more people began to listen. It was on an evening in Toronto when I realized just how courageous Jesse was, not just with his grit and determination, and with the knowledge that the future might not be all that great, that he knew who he was really helping. After completing our journey in Ottawa, there was now a new Canadian charity solidly in place and focused on funding research and making a difference for Duchenne families. Looking back, I had time to reflect on what had happened on the road, and I realized that Jesse had taught me more than I had ever taught him. While we made a good start in our ability to fund research, we still had a long way to go. And with that in mind, I took to the road again in 1998 to walk across Canada, to cement Jesse's journey as Canada's leader in funding research in Duchenne muscular dystrophy. This time I was alone in a journey that was going to involve parts of 2 winters. Starting in St. John's, Newfoundland, I spent 10 months on the road, most of it on the Trans-Canada Highway. It's probably worth noting here that in our time on the road in 1995 and in 1998, when I was crossing Canada, we did not have a website. Websites were in their infancy, and people didn't know they would one day be able to donate without using a check, a stamp, and an envelope. Day after day, province after province, I pushed on in both the heat and the rain, the snow and the cold. There was a particular day in Saskatchewan, and on that day, not one car stopped all day long. I felt that that was very much a test of character. It was one of the toughest days of my life, that 222nd day as I made my way across Saskatchewan. And the whole project was something that we did what seems like a long time ago, and yet at other times it seems like just yesterday. Jesse's journey has grown to become Defeat Duchenne Canada, and is making a positive difference for all those boys with Duchenne muscular dystrophy. We are now funding projects in countries around the world, a world which can thank a very courageous young boy who gave his all in trying to make a difference for others. The Duchenne research field has grown both in our ability to provide funding and in the number of researchers pursuing the cure that we know as parents is going to be found. This year, Canada ranks second in the world, behind only Switzerland, in the best countries on the planet to live in. Unfortunately, Canada ranks 17th on the World Index of Healthcare Innovation. Canadians endure the second highest drug costs in the world. Drugs that our sons need are available in the United States, the United Kingdom, the European Union, but not in Canada. This is the new frontier. This is where Defeat Duchenne Canada is focused on making a major change. There are many researchers who work tirelessly on behalf of your sons. Those researchers want to see a world without Duchenne, and we are honored today to have one of that dedicated pocket of world-class researchers join us this morning. Dr. Hugh McMillan is an associate professor in the Department of Pediatrics in the Faculty of Medicine at the University of Ottawa. He has a long list of credits, including being the author or co-author of 90 publications in peer-reviewed journals. He has recently been the senior editor of a publication that included contributions from more than 25 international contributors. Dr. McMillan has been the recipient of the Children's Hospital of Eastern Ontario Research Institute Outstanding Investigator Award and is a member of the Scientific Advisory Board of Muscular Dystrophy Canada. He has been the principal investigator for several Duchenne industry-sponsored clinical trials that Defeat Duchenne Canada is directly involved with. Over the years, I have seen many presentations, just like today. I have seen parents' eyes glaze over as they try to grasp the content of what is a very, very complex subject. And I've seen their eyes glass over as the subject has gone right over their heads. Over those years, we have made strides in asking researchers who are speaking to moms and dads who have no medical background to please, please, please do the best you can as a storyteller to create comparisons that we can relate to so that we can share your message with those people who are real philanthropists in the truest sense. They are the people who dig into their own pockets to help fund research. Dr. McMillan, one of the challenges today is to perhaps think of us as a group of grade 11 science students. We are grade 11 science students who are eager to learn and to carry your message forward. And on that note, Hugh, it's over to you.
Well, thank you very much, John, for that very kind introduction, and to everyone at Defeat Duchenne Canada for giving me an opportunity to speak today. Things have changed so much. When I think back, I was pretty young in the 80s, but in the 80s, through some of the advocacy efforts that people like John and others have done to fund clinical trials, to convince governments such as the US government to implement the Orphan Drug Act, it's really been what has stimulated a lot of the research that we're seeing now. In the 80s, there was no research being done on rare diseases. And the reason for that was because it was thought that one would never be able to make enough money from any of these products that any company would ever want to invest in them. And through advocacy and through fundraising, that has completely changed over decades. And I think that it's really the grassroots movements and convincing people in levels of government to make those changes, which has made the difference. I'm a pretty simple person, so I'm going to present this in a very simple way. I'd have a few disclosures of members of advisory board and some consultancy work that I've done for some sponsors. But we're really going to focus on two things. One, we're going to talk a bit about access to emerging therapies, clinical trials and approval pathways in Canada. And then we're going to review some promising therapies for DMD. And I'm going to present more concepts as opposed to granular data. I think some of the colleagues that will follow may be able to offer more in that regard. So with the scientific and regulatory pathway, there's many different stages to this. So it starts off on the extreme left with early research that's done in the lab. Then we move to doing work with animals. Then we move to clinical trials. And then after that, there's a Health Canada review that the industry will need to submit to Health Canada. And then it's reviewed following that by two different organizations, CADIT or in Quebec, INES, to make decisions about funding. And then it comes to market and is publicly funded for at least a subset of people with a given condition. And it's necessary to carry out post-marketing surveillance after that. The early research part is really basic science. So it's stuff that's happening in the lab with tissues, with molecules, and we don't tend to hear a lot about that. It's a little bit like what's underneath the tip of the iceberg that we see. And a lot of this work has been going on for decades. It answers things like, let's try to find a drug that can improve dystrophin production. Let's try to fit a gene inside a viral vector. Is it possible to do that? Can we make the viral vector get into muscle cells? So those are the sorts of things that happen in the lab. And then once there's a breakthrough there, it moves into preclinical research involving animals. And this will be important when we talk about some of the questions to make sure you discuss with the doctor who's running the clinical trial is what animals were studied. Mice are typically where we'll start, and then they'll move into dog models for dogs that have Duchenne muscular dystrophy and other animals that have models of Duchenne muscular dystrophy to be able to demonstrate that it's effective and to be able to demonstrate that it's safe. So questions that are asked at that stage are ones like, when we put the gene in the viral vector and we give it to the mouse who has Duchenne muscular dystrophy, how does it affect the mouse? Does the mouse get stronger? But it's really important because scientists have cured muscular dystrophy in mice dozens of times over. But it's important that those studies also move up and look at other, I would say, higher mammals for us to get a sense of how it's predicted to work in humans. So what animals were studied is a really key question. And then when one's talking about a clinical trial with their clinician, it's important to ask questions like, well, what benefits were observed in those animals? And what side effects were observed in those animals? Participating in a clinical trial, and I'll show you this in the next few slides, it doesn't necessarily mean immediate access to a drug. And we're going to talk about that when we talk about the trial design, concepts like randomization, placebo control. We'll touch on that in a moment. Important questions to ask. Clinical trials involve different phases. You've probably heard about, oh, it's a phase 1 trial, it's a phase 2 trial, it's a phase 2/3 trial. What those different phases mean is there are different stages at which those clinical trials are being carried out. All clinical trials share certain things in common. They involve human beings, and that's what differentiates them from the preclinical trials we spoke about before. They're prospective, meaning they move forward in time. So someone's given an intervention or a medication, and then we observe them moving forward to see how they respond to that. And it will compare an intervention, usually in the case of Duchenne muscular dystrophy, a drug or a gene therapy or a gene modifying therapy, to see how one group does relative to another, either a placebo group or comparing to external controls. And we have good external controls data now. They all have to be approved by Health Canada in order to be run in Canada. They all need to be performed at sites where there's trained and competent staff, and they all need to be approved by a research ethics board. Phase 1 studies are I would describe those as the early safety phase. Usually phase 1 studies are the first time a drug is being used in humans. For many drugs, phase 1 studies will be run on healthy volunteers. But for certain drugs like gene therapies, for example, it's not possible to do that. So it's run on a very small number of people who have DMD. The goals of phase 1 studies, and it's important to recognize this, they're about the first assessment of safety in humans. They're determining the safe dose range. And they're also about assessing how the body reacts to the drug, how the body breaks the drug down. So that's really the goal for phase 1 studies. One of the potential risks is the fact that it's the first in human studies. One of the potential benefits is they're usually run as an open label study. So there's not typically a placebo control aspect to those phase 1 studies. Phase 2 studies would be described more as an early efficacy phase. So it's giving the drug to a larger number of people with the disease. And in the cases of non-rare diseases, it's usually several hundred people. For the cases of rare diseases, it's anywhere in the range of about maybe 30 to 125 people. So we have to scale that down, of course. The purpose of that is this is the stage where we're really starting to see the first tests of possible effectiveness, of efficacy. And of course, there's ongoing safety studies as well. Phase 3 trials are even larger still. In non-rare disease populations, and I'm thinking about high blood pressure, estrogen replacement therapy, it usually will involve thousands of people. For rare diseases, it's in the range of about 100 people typically. And this is usually a placebo-controlled trial when we're at this stage. The goal is to really confirm effectiveness of therapy. And in Canada, for example, there needs to be a phase 3 element typically before a sponsor can move forward to asking for Health Canada approval. Phase 4 studies, we're not going to talk about in great detail because those are really after Health Canada has approved it. They're the post-marketing surveillance studies. And usually that's now done in the context of a registry for rare diseases. What you'll also see with rare diseases is increasingly there's a combination of the phases. So instead of it being a pure phase 1, pure phase 2, pure phase 3, phase 1 and 2 will be kind of rolled together. So there will be initial research that's being done about safety, but there will also be some efficacy measures that are enrolled in that as well. And then phase 2 will be a larger study, placebo controlled, phase 2, 3, and it will be run together. And part of that is recognize that there's a need to move quickly with these trials to make therapies available to people. And the other reason for that is that population numbers are low. So it can't be run like those old hypertension trials, giving antihypertensive drugs to thousands of people. It's not practical. The Health Canada review, the company, after they have the results of this data, and if it's favorable, then they make a submission to Health Canada to say, we would like to market this drug in Canada. The government will review that, and then they will provide an answer, yes or no, as to we agree to you marketing the drug in Canada and selling it in Canada. There's two possibilities. One is an accelerated review, and that's when there's determined in Canada to be an unmet medical need. And the review time for that is typically in the range of about 6 to 8 months. But a standard review, if there is an existing therapy already, can be much longer. It can be over a year. And what's frustrating about it is for the accelerated review, sometimes the payers will start to review it at the same time. But in other situations, we have to wait until after Health Canada has approved before CADTH or in Quebec, INES start to review in terms of should the provinces pay for it, for exactly whom should they cover, which adds to some additional delays that I'll talk about in a minute. Now, the US approval is a little bit different because they have something that we don't have in Canada. They have something called an accelerated approval program. And what that is there needs to be several things that are met. First, it has to be a drug or a treatment for a serious condition, and it has to fulfill an unmet medical need. But what's different about this is the US FDA will accept what's called a surrogate endpoint to allow companies to be able to put their product forward for potential approval. So examples of that surrogate endpoint that would be relevant to DMD would be serum CK. So if, for example, a drug or a gene therapy is used and it causes the serum CK to significantly drop, say to 10% what it was before, a company could file with the FDA for expedited approval or accelerated approval, but they can't do that in other jurisdictions like Canada or like the EMA. Typically, there needs to be more robust clinical evidence of improvement before it can be submitted to Health Canada or the European Medical Association in the EU. The companies still need to conduct the confirmatory phase 3 studies, or the US could pull the approval, but it does allow it to be able to become accessible much faster. And that's one of the reasons why we get frustrated in Canada, because we see these drugs available, as John mentioned, but we don't have access to them. And this is the part about the review afterwards. So even after Health Canada says, yes, we approve this drug, and that's usually the time where us as clinicians will have a lot of meetings with families to say, can I have access to this? Can I have access to this? And the frustrating part is until CADTH has done their review to determine whether or not it should be paid for and for whom with DMD it should be reimbursed publicly for, we can't access the money to pay for it for people who are on the OHIP Plus or the provincial payers in other provinces. And that can take time. Sometimes that can take a year or two for their review. Now, there's two exceptions to that. One is the first red dot on the left, and that's Health Canada's Special Access Program. A lot of people might be aware of that as it relates to accessing deflazacort. And it is a way where a doctor like myself can fill out paperwork and ask the government to allow a drug that's approved in another country or another jurisdiction to be brought into Canada for use for a specific person. And that's always something that we can apply to for a drug that's approved in the United States, but not in Canada. But the catch to this is that the cost of the drug can't be reimbursed. And that's a huge issue, especially with these newer treatments that are expensive. So it's difficult enough for families to be able to pay for deflazacort that we go through the Health Canada Special Access Program to get. But if we're talking about a drug that costs $600,000 a year, and we need to pay for that ongoing, or $2 million for a single treatment, that's just completely outside the reality of almost everybody. So the Health Canada Special Access Program exists, but it does not allow for reimbursement in any way. Because until it's approved by Health Canada, the government and the insurance providers, they don't consider it a drug. It's like me writing a prescription and saying, I'd like to import fairy dust. The government's not going to pay for that. And the insurance programs won't pay for that until it has a drug identification number, which comes with Health Canada approval. The second point here is after Health Canada has approved, and while we're waiting for CADTH or INES in Quebec to make their judgment about whether the provinces should pay, some people can access it at that point if they have private insurance. So I could write a letter and argue with an insurance company for them to cover the drug that's now approved by Health Canada under a private insurance plan to get access to it quickly after approval. But the provinces will not pay for it until after CADTH has made their ruling. There's two documents here if you want to read more about it, but they are a bit dense because it's typical government and related text. Now, some of the other considerations when one's talking about clinical trial design, and I go through all of this when I'm speaking to a person or a family about possible clinical trial participation, is ask about the trial design. Is this a placebo control study or is it an open label study? And the difference with that is placebo studies mean that there's a certain percentage of patients who will be randomized, meaning we don't know what they're getting, but they'll be randomized into getting active treatment with the study drug. And then a certain percentage of people will be randomized into a placebo, which looks like the drug, but it's not. So that's one thing. And if there is a placebo control, ask about what the randomization is. Is it 2-to-1? Is it 3-to-1? So you can understand a little bit more about the likelihood of being in one group versus another. What is the duration of the trial? Trials can run anywhere from a few months, most commonly a year, sometimes longer. And the duration of the trial might be significant depending on what your commitment is, how many visits you need to go to, and whether there is a placebo arm to the trial. A question is, and the answer is usually yes to this, but if my son receives placebo, will he get drug later? A very important question to ask. And will everybody get access to the therapy at the end of the trial? That should be yes, again, but it's still important to confirm that. What invasive procedures will be done? So are there sedations required? Are there muscle biopsies required? Just so we can weigh up again the commitment. And if that's something that is aligned with what your goals are. How frequent are the visits? How far do I have to travel? And is there travel reimbursement? There's a general, well, it's really accepted in the both from the sponsors and from the physicians running the trials that participating in clinical trials shouldn't cost you lots of money. So you should be reimbursed for travel, for your hotel stay as necessary. There's costs associated with taking time off work and just the stress of having to travel for it, particularly if you live out of city, but you shouldn't be paying extra on top of that to participate. So some of the promising trials, I'm going to shift gears a little bit. And one of the questions that is very important to ask whenever you're hearing about a trial or you're hearing about a treatment, in the case of DMD, is to ask yourself, is this specific treatment specific to a DMD mutation or not? So there's some treatments on the right, like the exon skipping therapies, where an individual has to have a deletion of a specific area for them to be a candidate to potentially benefit from that therapy. And then there's others on the left, which I'll talk very briefly about each of them, where you don't need to have a specific mutation. They're potentially beneficial to all patients with DMD. So prednisone has been around since the 70s. We've known that it has offered some modest benefit since that time. We know that the goal of prednisone is to try to prevent this trajectory from left to right, where we start off with these beautiful circular muscle fibers, where with time there's these little tears that we start to see. And then as we move further to the right, there's less muscle fibers and there's more fat and there's more fibrotic tissue. So we want to find therapies that ideally stop this progression and at the very least slow it down. One of the studies that I'm going to talk about, one of the products that I'm going to talk about is vamorolone, which is an alternative to prednisone, an alternative to prednisolone or deflazacort, which were some of the more traditional corticosteroids that you might have heard about. And vamorolone is a steroid. It's a synthetic dissociative steroid. And you'll hear more about this a bit later today from Eric Hoffman. But there's a couple changes on the molecule that you can see by the red arrows on the right that make it different from prednisone. And there was a very important pivotal clinical trial that has been published, and it looked at 121 boys with DMD that were between the ages of 4 and 7. And for the first 6 months of the trial, they were randomized into low dose or high dose vamorolone, standard dose prednisone, or placebo for the first 6 months. And what was very impressive with this is the vamorolone met all of its primary and secondary endpoints, meaning that the different motor tests that were being studied to see whether the drug appeared to be effective or not were uniformly met, which was quite impressive. We don't tend to see that in all clinical trials. Examples of that, and I'm only showing you one slide on their primary endpoint here. When we look at the duration of the trial on the bottom axis, on the horizontal one. And then if we look at the change in the time to stand, so how quickly the boys could get up from the floor, when the placebo is the gray one at the bottom. So there was a flattening and then a decline on the time to stand. But we could see for the vamorolone treated boys, both at the low dose and the high dose, they did significantly better on the time to stand score. And we saw that on other outcome measures as well. It also showed a much more favorable side effect profile to the prednisone as well, which was very, very encouraging. And as a result of that, it's starting to emerge as a potential alternative. We do know now that 6 milligrams per kilogram per day is really the best dose in order to preserve motor outcomes. Lower doses are not as effective and potentially can have other complications, including adrenal problems, which you heard about yesterday. One of the important things is linear growth appears to be spared with vamorolone. And that's an important difference, as we all know, compared to prednisone and deflazacort, where particularly deflazacort, there can really just be a plateauing or a flattening of linear growth height when that's started. Weight gain is seen, but it might be a bit less obvious if an individual is continuing to grow. That needs to be monitored. Some of the early studies found that early bone markers were also favorable, but as Dr. Ward mentioned yesterday, fractures can still occur. We don't yet know a lot of the long-term effects on bone, muscle, puberty, and that needs to still be studied. There's ongoing clinical trials with vamorolone that are happening here in Ottawa and in many other centers around Canada, looking at very young patients that are between 2 to 4, and that's still recruiting, that are steroid naive, as well as boys who had been on prednisone or deflazacort between the age of 7 to 18 that switched over to vamorolone. And those studies are full, but they're ongoing, and we hope to have data from that very, very soon. There's also some studies for young men with Becker muscular dystrophy to see if it's effective in that population as well. So there's some encouragement from that. Givinostat is another one that you may have heard of, and I think we're getting a talk on this a little bit later today as well from the sponsor at Tal Pharmaco. And Givinostat is a small molecule that's called an HDAC inhibitor, histone deacetylase inhibitor. And some of the preclinical studies that were done on mice showed very encouraging results in that their muscles had less fibrosis and their muscles were also larger compared to untreated mice. And what was very encouraging is there was a phase 3 clinical trial that Tal Pharmaco ran, and it had very, very positive results. The 179 boys with DMD who were randomized in this trial were followed for 18 months, and they showed a positive primary endpoint, meaning that they had an improvement in their time to climb up 4 stairs, The decline for the boys who were treated with givinostat was much less than what was seen for the boys who were in the placebo group of that trial. The secondary endpoint looked at MRI, and I'm just showing you an example of that at the bottom. They looked at one particular muscle called the vastus lateralis, which is part of our quadricep muscles, and they found that the givinostat group showed less fibrosis, less abnormality there compared to the group that received placebo. So very encouraging results from that as well. And there's some ongoing clinical trials for younger and also older young men with DMD that will be starting soon with givinostat. Another sponsor, Edgewise Therapeutics, is carrying out a clinical trial in both Duchenne and Becker muscular dystrophy. And they're looking at a small molecule that's designed to protect the muscle from tearing by modulating one of the very, very important proteins that are involved in muscle contraction called myosin. So that's a trial that's ongoing, and we'll be quite eager to hear the results from that as it becomes available. Ataluren is one that you may have heard of from PTC Therapeutics. Ataluren is an example of a drug that is mutation specific. So Ataluren is specific or of potential use for people that have what's called a nonsense mutation. And that's where there's a spelling mistake in the DMD gene that causes the body to stop reading the gene at a certain point. So this is not a gene deletion. It doesn't help with that. It does not help with duplications. It only helps if there's a spelling mistake and the body stops reading it. Ataluren helps to force the machinery to continue to read the gene and to give more of the full-length normal dystrophin. So it's helpful in that population. There was a study that was done in 2014 that showed an almost, but not quite statistically significant difference between the placebo group and the children that received 10 milligram per kilogram in the morning, 10 milligram per kilogram midday, and 20 milligram per kilogram at the end of the day. And there's a much larger 18-month study that was just completed. So we're hoping to get the results of that very soon. And if it's positive, it may be an option for people with DMD who have nonsense mutation. So keep an ear out for that, because we are expecting to hear those results in the very near future. Now, gene therapies, I'm going to talk about these in very broad terms, but I like to break them down into two big categories. The first are gene modifying therapies. Those are the ones that function on RNA, and I'm going to explain to you in a minute what RNA is. They alter the way the RNA gets put together. We also talk about them as exon skipping therapies. Okay, so I'm going to give you an overview about what those are, and it will hopefully help with some of the data that's going to be presented a little bit later to understand and interpret that. The second broad category are gene replacement therapies. So those are ones where typically a viral vector is used, an adeno-associated virus. A part of the gene, a microdystrophin or a mini dystrophin, is packaged inside that viral vector, and it's given back through an intravenous dose, finds its way into the muscle, and the hope is that it does the job that dystrophin would have done to keep the muscle strong. And we'll talk about that as well. So first, a very basic overview of DNA to RNA to protein. And I like to kind of find an analogy to help me understand what the heck these differences are. DNA is really like the master blueprint. All of our body's cells have one copy of that master blueprint that tells our body's cells how they should run, how they should be made, how they should do all of their jobs. And because it's so precious, it's kept in the nucleus, which is sort of a protective area of the cell to prevent it from getting damaged. Now, the RNA is like a photocopy of the blueprint that gets made and gets sent out to the work site. Because you need to be able to look at the blueprint when you're at the work site to figure out what to do, how to build things. So there's lots of photocopies that are made, and then that RNA gets sent out into the cytoplasm outside the nucleus for it to then be used to make proteins. And the proteins are like the construction workers that carry out the day-to-day work. So the gene modifying therapies that we're going to be talking about are actually working on the RNA in the middle. They're working on those blueprint copies that are going to be sent out to the work site. The gene replacement therapies, which we'll talk about later, are helping to replace a version of the master copy that's in the case of DMD is damaged. The gene therapy pipeline in terms of gene replacement therapy, the RNA therapies, which are the gene modifying, or even cell therapies for all diseases, not just DMD, has been exploding over the last decade. And this is a graph showing you each bar is each year. The most recent one on the right is 2022. And there's over 2,000 different products that are at various stages of preclinical testing and clinical trials. It's really quite remarkable. Most of it's being driven by the preclinical work, which is the bottom sort of darker purple, but you can see from the lighter purple and the orange color, there's more and more that are coming into clinical trials as well. Now, to look at the DMD gene, I think of it as pieces of a puzzle. There's 79 different exons or 79 different puzzle pieces that we all should have in order to have normal dystrophin. And all those puzzle pieces have to be assembled. So when we make that blueprint and it's being sent out to the work site, all of the pieces of that blueprint have to fit together in order for dystrophin to be made and for that dystrophin protein to do its job. And it's a big, big, big gene. And that's going to create some problems for replacing that gene, which we'll see in a moment. But this is just a hypothetical example of a young man who has a deletion of exon 50. And what that does is if you look down to puzzle piece 50, you can see that the two puzzle pieces on either end, puzzle piece 49 and 51, they can't fit together. Those two little shapes can't be made to fit together. And because of that, that's what we call an out of frame mutation. And when that happens, all of the pieces get broken down. Now, exon 51 skipping therapy, for example, is designed to try to help with that. So you can see above puzzle piece 51, there's this little tiny, almost looks like a comb, sort of an old school comb, and it binds to exon 51. And then that allows 49 and 52 to now fit together. So if you see that, it's taken what was previously what's called an out of frame mutation by silencing or suppressing that exon 51. Now 49 and 52 can fit. And when that happens, that's now called an in frame mutation. And that's what we typically see with the milder version, Becker muscular dystrophy. So that's really the goal of exon skipping therapy. And they can target different puzzle pieces depending on the different mutations that people have to try to bring those out of frame mutations back to being in frame and allowing for some dystrophin to be produced. That's the goal. Now, this is a bit of the discrepancy that John was talking about earlier. And this is the reality, the frustrating reality that we live in, is that the US, for example, has 4 separate exon skipping therapies that have been approved and have been approved for some time. For exon 51 skipping therapy, there's the Teplersen. For individuals who have deletions that could be fixed by the exon 45 skipper, Casimersen. And in the US, there's 2 exon 53 skippers that are approved, Golodirsen from a US company and Vitala from a Japanese company. None of these are approved in Health Canada. About 60 to 70% of boys who have DMD will have an exon deletion. Of those boys, about 30% of them could benefit from treatment with one of these 3 different strategies. And overall, if we think about all of those boys and young men who have a deletion, almost 80% of them could be treated with an exon skipping therapy. The exceptions might be people that have incredibly large deletions, but even then, I think there's still a possibility. And there's even some potential for antisense oligonucleotides to be used on people who have exon duplications as well. It's not unique to deletions, but that is the area of focus for a lot of the research that has been done so far. Now, there is something called the next generation antisense oligonucleotides, and you might have heard of this. There's 5 companies that are working quite actively in this field. The next generation antisense oligonucleotides, they link the exon skipper, which is also called an antisense oligonucleotide, to a protein to allow it to have better penetration into the muscle tissue. Another strategy that Wave Life Sciences has is to alter the shape, or we call it the stereochemistry, to potentially allow it to have better entry into the muscle tissue. Because in order for the exon skipper to do its job, it needs to get into the muscle in enough quantity for it to actually start to cause that skipping to take place. And some of the preclinical studies have shown that these new or next generation antisense oligonucleotides can have up to a 10-fold increase in inducing exon skipping and dystrophin production compared to some of the traditional antisense oligonucleotides for Duchenne muscular dystrophy. And even more than that, there's the potential for reduced frequency of dosing. So and I say older, it's a bit funny to say that because they're pretty new, but the older antisense oligonucleotides need to be given every week, but these new next generation ASOs potentially can be given every 4 weeks. And those are actively moving into clinical trials right now, some of them ongoing, and you'll hear more about this later today. In terms of gene replacement therapies, so these are the ones going back to the DNA. I had showed you an earlier slide about really the explosion, about the research that's going on, preclinical and clinical research. These are examples of approved gene replacement therapies. These are where DNA is packaged up into a virus, that virus is given to an individual, and then the gene, or in the case of DMD, part of the gene, it finds its way into the target tissue and starts to do the job that the original gene should have done. Now, there's 26 different gene replacement therapies that have been approved in at least one country. If I was to put little American flags besides these, most of these are approved in the US, not all of them. In Canada, we have 4 approved therapies for different types of cancers that affect adults. And then there's 2 for various neurological conditions. Omnisemnogene abeparvovec is probably the best known, or it's also called Zolgensma for SMA. And then there's another that can be used for a form of congenital blindness. That product's called Luxturna. You'll see at the very bottom that DMD is approved in the United States, the gene replacement therapy from Sarepta and Roche. And it is approved for boys who are 4 to 5 years of age under their expedited approval process. And I will talk about this a bit later. Now, one of the challenges that we face with Duchenne muscular dystrophy when it comes to gene replacement therapy is, as I mentioned before, the DMD gene is huge, really big. It's about 2.6 megabytes. So if we think about our little flash drives, it's like one of those sticks we put in. Now, AAV vectors can only carry about 4.5 kilobytes of genetic information. So think those, and some of the younger people in the room will have no idea what I'm talking about, but think about those 5.25-inch floppy drives that we used to stick into our old computers. That's sort of the capacity of those AAV vectors, but we want to put the information that needs to go on one of those fancier flash drives. Another way to look at it is if we think of that stack of papers there, if I was to say to someone, okay, I want you to fit that into the envelope on the right, you'd look at me and you'd be like, yeah, okay, it's not going to work. So how do we make this work? So there was a very good observation, and a lot of what we learn comes from being aware of the people that we care for. We learn about things in so many ways for the people we care for. But there was a group of individuals that worked in the UK that followed a family that had a deletion within their gene that took out basically half the DMD gene. So this family had a deletion of exon 17 to exon 48. But what was really interesting with this is it was an in-frame mutation. So you can see that exon or puzzle piece 16 can fit to puzzle piece 49. And what was reported with this family is despite having lost about half of the DMD gene, these people did not show any symptoms. The men that carried this change in their DMD gene did not show any symptoms until they were in their 30s, and they were walking well into their 60s. So it kind of raised an eyebrow, and it said, man, you can actually do really well with very little gene. So that sort of opened the idea to this whole concept of mini dystrophins and microdystrophins. So you see on the top there a figure that represents what a quote normal dystrophin gene would look like. And then I'm giving you two examples. The one on the top is from Pfizer. The one on the bottom is from Sarepta and Roche. And it's showing a promoter in yellow to sort of kickstart or help with the production of that gene. And then pieces of the gene, but you can see it's much shorter than what we would expect to see in a typical dystrophin gene. But because there's less there, it can be packaged up into that viral vector. Now, I'm going to show you some results from the Sarepta and Roche trial, and I'm not favoring one product over the other. I'm showing you this data just because the work that's been done with Sarepta is a bit further ahead, and there's more that's available in the scientific literature and in the public domain. The other trials are ongoing and will be reported out hopefully very soon. And I'm thinking specifically of Pfizer and Solid Biosciences, but they're not available in the public domain yet. But some of the very encouraging results that we see from the Sarepta and Roche product, and I do think there's a competition to make these names more and more difficult to say. But the gene therapy that they offer, delandistrogene moxeparvovec, one of the things that was found with this was when they give the individuals with DMD the gene replacement, first, does it get where it needs to be? So they looked at that in two different ways. One is something called the Western blot, which is a way of taking the muscle and basically filtering out just the dystrophin and then asking if there's evidence that there's some mini dystrophin or microdystrophin that's in the muscle fibers. And there was. You can see the little blue arrow. It's a little small. There's no line there. That's the baseline study. And then the same individual was checked again 12 weeks later. And you can see now there is a little band there where the red arrow is. So there is evidence of dystrophin getting to where it needs to be. And that can be checked in another way too, something called immunohistochemistry, where a muscle biopsy is done and then staining is made on the muscle to again ask the question whether there's dystrophin getting to the muscle. So what we see in these four little colored pictures is the picture on the upper left is an individual who does not have Duchenne. And you can see all those red circles. Those are all those muscle fibers showing dystrophin in the membrane. On the top right is an individual at baseline, and it's black because there's no dystrophin there. It's all been broken down. On the bottom two slides, we see two individuals that were treated with gene replacement therapy, and you can see evidence of those red circles there. So the dystrophin is getting to where it needs to be. The second piece to this then is it having a clinical benefit? And this is where things get a little bit more murky. I'm showing you this just because it helps to understand a little bit about why the US FDA approved the gene replacement therapy for the 4 to 5-year-olds, but not the 6 to 7-year-olds. What it showed was when they compared the boys who were treated with gene therapy in the solid black line, compared to a placebo group, which is the dotted line, and this is on the left, the 4 to 5-year-old boys, there was a significant difference there. When the 6 to 7-year-olds were looked at on the right slide, there was not a significant difference there. And it actually is, at first glance, it's hard to actually interpret what the heck it means because we see the placebo group actually being higher than the treated group. And the reason for this is there was a problem with the trial design or the randomization process where when they were shuffling the cards and randomizing some boys into placebo and some boys into the treatment group, they randomly shuffled the stronger boys who had better timed functional scores into the placebo group, and they randomly shuffled more of the weaker boys into the treatment group. So it's hard to interpret that because of that problem with randomization. And we do need more data to understand better what's going on in those older boys. And on that basis, the US FDA said, well, we see clear evidence of the 4 to 5-year-olds. We will approve it for that, but we want more evidence for the 6 to 7-year-olds. And there has been some motor outcome results that have been forwarded, including the Embark study results most recently. And the results were a little bit confusing. So I explained to you already in the top part, that's the SRP 901-102 trial. That's the one where I showed you that there was improvement in the 4 to 5-year-olds, but not in the 6 to 7-year-olds. The other challenge that's come up was the recent results from the Embark study, which studied the same age group from 4 to 8. It was a phase 3 trial, and you can see there was 125 boys in that trial. The primary outcome was a timed functional test that many of you are probably quite familiar with, called the North Star Ambulatory Assessment. And it's a timed functional test measure that will ask 17 different questions. So things like, how does the person with DMD stand? Can they stand with their feet flat on the floor, or are they up on their toes? And there's different scores given for things like that. How well can they step up on a step? How well can they step down from a step? So it looks at 17 different questions and assesses a score based on their performance on that. That outcome measure was not different between the placebo and the treated group. But 2 of the other timed functional tests were different. The time it took for those boys to get up from the floor and the time it took for those boys to walk 10 meters was significantly better in the boys treated with gene therapy compared to the boys who received placebo. And that was very encouraging. And I think it's going to take a little bit of time to unpack this, to try to understand what all of this means, and probably to get some additional information and longer term information to be able to interpret this a little bit better. I personally think there's some limitations to the North Star Ambulatory Assessment in this age group, because we're seeing a test where some of the boys will be improving, especially the 4 to 5-year-olds, and some of the boys will be declining, especially the 6 to 7-year-olds. So we're measuring a placebo and a control, and some of the population's getting better, some are getting worse. It's a really mixed bag. So I'm not convinced personally, and I didn't design the trial, and they probably would not welcome my comments now that it's been finished. But I do have some reservations about the use of the North Star in this specific age group for this specific trial duration. But it is what it is, and it'll need to be unpackaged, and we'll need more and longer data, and we'll also need data from Pfizer and Solid and some of these other companies that are running trials in parallel for us to get a better understanding as to whether this is or is not a therapy that will move forward in the long term. So I'm going to stop there. The conclusions from this is I really hope that I've kind of given you some additional questions in your toolbox that you can use to try to organize whether or not what you should ask basically when you're considering a clinical trial, and to really reassure you that there's some promising therapies for DMD that are here, and there's some other promising therapies that are on the very near future. And this is a huge change in landscape from what we were discussing and what we were thinking about 10 years ago. So we are living in a time of tremendous optimism, and myself as an individual who is fortunate enough to provide care for people with DMD and their families, I'm very, very excited about some of the opportunities that are coming. And we may be in a reality like people with SMA are, where we're not just talking about the one therapy that might work, but we're talking about multiple beneficial therapies that are approved, and which one of these therapies might be better for the person that's in front of me. But don't stop the fight. You know, we do need to push the government. We do need to push the regulatory bodies. We do need to ensure that things continue to move forward, because as we've seen, there can be some significant delays, some significant discrepancies from country to country if we take our foot off the gas. So I'm going to stop there, and I think there's lots of time for questions, but the moderator can pull me off with a cane if I run over time. Thank you. Ton of information. Yeah.
Or just going to ask one question.
Yeah, of course.
In terms of, you know, how far behind Canada seems to be to the US in approvals and whatnot, is is most of that because of Health Canada, or is it because the drug companies just are not moving forward with the filing within Canada? How would you break those down in terms of why that discrepancy might exist?
Yeah, I think it's multifactorial. And I think that I think there's several obstacles. So I think it's, in my opinion, and this is my personal opinion, I'm going to preface it with that. I think in some ways the obstacle is less with Health Canada than it can be with CADIF in an S that come afterwards, because the entire process is quite time consuming and quite laborious. And for some countries, and I'm thinking about countries in Europe, for example, when they achieve regulatory approval from their national regulatory agency, bam, the drug is available. Whereas in Canada, man, that is not the case. That is the first step in a long fight to be able to get access to drugs. And CADIF, and again, this is just my personal feeling and my personal experience, can be a challenge. It's a review process. And if anyone from CADIF is here, I'm happy to engage with you. But it's a review process that takes a year or two. And then my experience with other neuromuscular diseases, at the end of that 1 or 2 year wait, what they've basically done is they've just regurgitated the inclusion-exclusion criteria of the pivotal clinical trial. So I don't understand why it takes that long for them to then just regurgitate that, we will approve the product for individuals who, are within this age group and within this functional status, so it's very, very similar to what the inclusion-exclusion criteria of the trial were. And I think as a clinician, one needs to think more broadly. So it's not enough to just say, oh, you studied it in patients that were up to 8 years of age, or within that 4 to 8 year window. And this is, again, assuming that it's effective and that there's evidence of effectiveness. But it's also important to say, well, let's take a step back and say, from a theoretical perspective, well, why would we not give it to a 3.5-year-old? Or why would we not give it to a child that's 8 years and 1 month old, if we would give it to a child who's 7 years and 11 months old? So there's things that boggle my mind about, where do you draw these firm lines in terms of cutting people off for funding for a product that don't make logical sense to me. Not everybody with Duchenne is the same, and this is the same for other neuromuscular disorders. So there needs to be some case-by-case flexibility with approval for reimbursement. So all that to say, to answer your point, I think one of the big challenges can be that companies know that they're up against significant hurdles, significant expenses, significant durations of time to be able to not just achieve the Health Canada approval and everything that goes with that, to then only start the whole process again with the second independent group that's now going to talk about reimbursement and who should receive it, who should be funded to receive it. So that's part of it. I think the market in Canada is also smaller. So I think that can be a disincentive for some companies to move forward and have a Health Canada submission. So they need to think about, and I'm sure the sponsors will think about, how many people could potentially benefit from it if the drug costs X number of dollars, what would be the profit versus the expenses associated with Health Canada and CADIF approval. So I think there's multiple different factors there that can influence a company's decision to file or not file with Health Canada, and certainly an incentive for them to file for other countries first that don't have some of the same challenges that we have. I'm not sure if I've answered your question, but it's bloody complicated.
No, for sure. But is there no collaboration whatsoever between the regulatory bodies in the different jurisdictions then, between FDA and Health Canada? Do they not talk to each other whatsoever? Is there how do we get to a point where a Health Canada, for example, will say, well, the drug is approved there. They've obviously gone through all the checks and balances, similar to, an airplane getting approved to fly in the US versus in Canada, Transport Canada versus. So is there any movement in that direction, or is that the pipe dream?
I think there needs to be more of that. And I think that's an example of where we could advocate to try to move things forward. And this isn't unique to DMD. I think this is a good example of a fight that could be taken on with other parent organizations from other rare diseases to try to unite and try to push for some of these regulatory changes. Because I agree with you, if a product has been reviewed already by the FDA and has been approved by the European Medical Association, what are we going to come up with? They do tend to always go back to the thalidomide argument, because there was, it was before my time, but I do know that thalidomide was approved in the US, and it was either not approved or there were delays in approving it in Canada, and that will often be held up as the reason why there needs to be an independent review. But I would argue that I agree with your point, and I feel that there needs to be some push towards streamlining the process, having the CADITH review, if it even happens at all, happen at the same time as Health Canada, to be part of that process, as opposed to a separate and a secondary review, which is nothing short of a time waste to wait, because there's nothing more frustrating than when Health Canada has approved it, and despite that, I can't access it, and my families can't access it. It's just, it's mind numbing. It's bad enough when it's available in the US and not in Canada. That's difficult enough for all of us, but it's even worse when it's approved by Health Canada and we're waiting a year, 2 years or longer to hear from CADITH in the province, in each individual province, if they're going to pay for it or not. And if they do pay for it, for which subset of patients with DMD. So that's an important area for advocacy. And I think now is a good time for that.
Thank you.
Thank you.
We do have a question online. What can or is the medical community willing to do in conjunction with rare disease groups to help accelerate the process and create a more welcoming approval system?
Yeah, I think we share the same frustrations that families have, and individuals like myself, and certainly other not just pediatric neurologists and neuromuscular folk, but really people who work with individuals who have rare diseases. We're happy to stand up and help advocate, but the changes have to come at the regulatory level. These changes have to come at Health Canada, and then at the provincial level, where they rely on CADITH to make these decisions. And I think that advocacy work needs to come from organizations like this. We're happy to back you up and to speak, but politicians care more about what their constituents want and think, and care a whole lot less about what I think. So I think that there's strength in advocacy, there's strength in grassroots movements. We're happy to lend a voice to that and speak very loudly, but I do think that some of these changes really need to take place at a grassroots level.
Thank you, Dr. McMillan, for this great presentation. As a pharmacist, I really enjoyed listening to it, and I learned a lot. So my question is, sorry if it's a simple question.
No.
Has there been any attempt to clone the dystrophin protein like an antibody? And even it is possible to create a protein similar to dystrophin and have an injection for that?
Yeah, there have been some studies that have looked at analogous proteins. So a target for a long time was, there's like a sister to dystrophin, something called utrophin. And utrophin is actually expressed when babies are inside their mums. And then after babies are born, there's a switch over from, well, even just before they're born, there's a switch over from utrophin to dystrophin. So one of the therapies was to try to aimed at trying to increase the amount of utrophin as a bit of a surrogate for Duchenne, to kind of compensate for the problem with dystrophin. There hasn't been any real breakthrough therapies with that per se, but that has been an active area of research. And a lot of the challenge with dystrophin, the protein, And dystrophin, the gene, is its size, its large size, and getting the gene or the copies of the gene, the RNA, to where we need it to be. It's been a challenge, and there's fortunately been some strategies on multiple angles, as I showed you, gene replacement, gene modifying therapies, to be able to try to counter that, as well as some of the small molecules, which I talked about earlier, and they really target the protein to kind of help the workers at the construction site to be able to do their job better. When we talk about things like vamorolone, or we talk about other agents too, like the study with Edgewise, it's a little bit like giving the construction workers better boots and better tools so they can do their job more effectively. So they're all aimed at different parts of the process, but the size of the dystrophin gene has been the biggest challenge.
One more from online. As the data unfolds for the EMBARK study, is it possible for the authorities to approve the gene therapy despite the primary endpoint not being met?
I think it's possible. I think it will require, I use the term sort of unpacking the data. I think it will require some interpretation about what do those results mean, because it's very simple when we see a trial where the primary outcome measure is positive. It's also very clear when the primary and the secondary outcome results are all negative. But where it gets more difficult is when we see a situation like this where the primary outcome is negative and we see two very impressive results with the secondary outcomes. So what does that mean? Does it mean that maybe the primary outcome measure shouldn't have been the primary outcome measure? And that the other secondary outcome measures were actually telling a better story? Or does it mean that the primary outcome was negative and therefore the product is not beneficial? And I think that's, with that unpacking and trying to understand that, that's what will need to be interpreted to decide if the company does want to move forward for approval process. And we'll have to wait and see what their interpretation of the data is and how they feel is the best path forward. It's encouraging, and I think it's really easy to sort of capture things in a little news snippet. And when I was preparing the talk, I was reading about it, and a lot of the news releases were like primary outcome negative. And when I read it at first, I'm like, oh man, that really sucks. And then when I read it in more detail and I read the full press release, I'm like, wait a minute, the primary outcome is negative, but these secondary outcomes are impressively positive. So I think we have to push past just the quick one-liners or the headlines we read on the news and interpret the whole thing. And we'll have to wait to see what Sarepta and how Roche interpret this moving forward.
Great presentation. Just a couple of questions I have. The first one being with the new generation of exon skipping coming up, right? And some of the new drugs like vamorolone or Givinostat being kind of approved in the other jurisdictions, right? Are you as a physician and other physicians in Canada working closely with these new exon skipping pharmaceuticals to kind of have these new drugs as an inclusion criteria for the therapies, right? Right now, my understanding is that if somebody is on vamorolone or some of these drugs, then they are excluded from some of these exon skipping therapies. And what are we doing to make sure that those individuals are included, and how are we gonna proceed moving forward with this?
Yeah, thank you for that question. I think it's always a challenge because with clinical trials, there's typically an exclusion criteria for an individual who has received another investigational product, or who is receiving sometimes another investigational product. And they might allow someone to participate after an appropriate washout period. But the rationale for that in most clinical trials is because they want to try to control things as much as possible, and they don't want this other variable floating around that might make a trial negative that could have been positive, for example, that might dilute the potential benefits associated with the product that's being studied. For most clinical trials right now, I would actually say for all clinical trials right now, vamorolone would be considered an exclusion criteria at this point in time. It would be necessary for an individual who's on it to switch over to prednisone if they wanted to receive a gene replacement therapy, or switch over to deflazacort or prednisone if they wanted to be on an exon skipping therapy. But I think That is likely to change, particularly now that vamorolone has been approved by the US FDA. Each of the individual sponsors that are running different trials for different products will have to weigh up how much is vamorolone now part of the landscape. Do we know enough about it that we feel it's equivalent to prednisone and deflazacort in the sense that it wouldn't contaminate the trial or wash out a potential benefit or non-benefit from the placebo that might make it harder to interpret the data. So it will come, it has to come, because whenever something is part of standard of care and approved, that needs to be reflected in the trial design. But we're not quite there yet. We're moving there.
But what can we do to kind of, like make sure these pharmaceutical companies do understand that this will become the standard of care in the future, right? With the data that we have, right? And it's not like within the Duchenne communities, our kids have a lot of time. It's all time sensitive, time based, right? So what can we do to kind of, progress that?
Yeah, I would say talk to the companies. I do know that there are discussions that are already underway about the changing landscape with vamorolone. So the companies are also, I'd have to say, they're all very cutting edge with this. Like they're constantly seeing changes in terms of treatment. They'll revise their protocols frequently, either based on new safety signals or other drugs that become approved. So they're very well aware of this. And I would say that I predict that vamorolone will find its way into various protocols in the not too distant future. But it also needs to be approved in order for it to be part of standard of care. So it's approved in the US, and trials that are US based or US centric, I think, will be forced to adapt to this very quickly. But for us as Canadians, we need to see some of these new products approved for them to be part of our reality.
Go ahead.
Okay, so my question is in regards to Casmirsen,
Yep.
for instance, and the Sarepta clinical trial. If the medication is approved in the States, but is still in clinical trial in Canada, can the special request be done for children here that would benefit from that medication be done?
Yeah, so the quick answer is yes. There could be an SAP application for Casmirsen to be given on clinical grounds for an individual who has DMD who may benefit from exon 45 skipping, but the challenge is paying for it. That's the big issue.
I was going to ask.
The process would be, me filling out about a 4 to 5 page set of paperwork and then asking the government to allow me to import Casmirsen to my pharmacy to be given to Bob Jones, my patient. But Bob Jones and their family would have to find the money to pay for that Casmirsen because it's not a drug in Canada, even though it's approved elsewhere. So it doesn't have a drug identification number. So the provincial government won't pay for it. The private insurers won't pay for it. And it's expensive.
What is it running at?
I think it's in the ballpark, and I'd have to talk to my pharmacist to give me a more accurate price. But I think it's in the ballpark of like $400,000 to $500,000 a year.
A year?
Yeah, that's the ballpark of it. So the clinical trials have completed for Casmirsen. Like they finished enrolling, and the young men are rolling out of that trial. My hope is that Sarepta will file for approval. They haven't.
Mm-hmm.
But another consideration would be, are there other trials, these next generation exon skipping therapies that will skip exon 45 that might be a potential? And I have to look at the inclusion exclusion criteria to be able to, to guide specific people with regards to potential trial eligibility. There is a really good website, www.clinicaltrials.gov. It's a clinical trial site that's basically run by the US National Institute of Health, and sponsors need to post on that if they're running a trial, and it will list all the key inclusion exclusion criteria, and you could look at that to see if it looks as an potential and initial match. So I would really encourage people to look at that from time to time.
Okay, we just have two more questions at this table and then we'll wrap up.
Sure.
Thank you. I was wondering, Dr. McMillan, about the gene therapy. You identified that technically it should be working on pretty much all the boys, but I was wondering if they're in the trials, either Pfizer, Sarepta, Roche, if there's some limitations that have been identified. For example, location of the deletion, or the size, or if it's in frame, out of frame, or yeah.
Yeah, no, it's the big group that have been excluded from gene replacement therapy participation at this time are individuals who have exon deletions that affect the earlier part of the DMD gene. So the reason for that, and there was a very good article that was just published in the New England Journal of Medicine about this. Carson Bonneman was the first author, but there seems to be a higher risk of complications to gene replacement therapy if an individual has an exon deletion that's very early in the gene. So just because of that, and for safety considerations at this point in time, that's one of the exclusion criteria. But that may change with time. And there's other strategies that are being looked at, but that is one of the exclusions right now.
Thank you.
Perfect. And I think that's all the questions in the room now.
Excellent. Thank you all for your attention.